Session of the Wales Biodiversity Partnership Conference, the Tuesday afternoon session. My name is Ant Rogers and I'm the Biodiversity Implementation Officer for the Pembrokeshire Nature Partnership, one of the local nature partnerships that works throughout Wales to coordinate and promote actions to facilitate nature recovery in Wales. This afternoon session is all about tracking biodiversity, distribution and trends. So we've got a series of really nice talks for you. Mostly looking at how we collect and use data in our work. You can ask questions throughout the session. If you use the live chat facility, you can type them in there and we will try and pick up as many of those as we can after the speakers have finished. We'll have two presentations and then a Q&A session, a break, and then repeat the whole process again. So feel free to type your questions in the live chat there. The speakers' biographies are all available on the main platform website. As is the feedback facility, and the organisers would really like it if you could give feedback on the session so that they can try and improve things for future iterations. The sessions are being recorded as well, just so that you know. And for those of you who are socially minded, just to remind you that there is 6 o'clock tonight, a social networking quiz on the platform, details on the main page again. So without further ado, we will start with our first presentation from Eleanor Colver of the British Dragonfly Society. Over to you, Eleanor.
Everyone, Eleanor Colver here from the British Dragonfly Society. I'm going to be talking about the results of our Clubtail Count citizen science project, which ran from 2017 through to 2019. So for those of you who aren't familiar with the common clubtail, its scientific name is Gomphus fulgatissimus. It's quite a small dragonfly, only about 5 centimetres long, and it has quite an unusual characteristic. As you can see in this photo, it's got bright green eyes, but they don't meet in the middle of the head as they do in most other dragonfly species. So that's quite a defining characteristic. Here we have a female, and as you can see, it's got bright, jazzy yellow and black markings. Here we have a male, quite similar in appearance to the female, but with more of a club-shaped abdomen. As it matures, it takes on more of a lime green colour rather than the bright yellow of the female. And if you have a look at the base of the hind wing, you'll notice that there's a little notch right at the base, and this is a way, again, to separate males and females. So adults spend most of their time in deciduous woodland, foraging for other flying insects to eat. Often they'll actually mate away from the water as well, and then the female will return to water to lay her eggs. And their preferred breeding ground is slow-flowing lowland rivers. Here we have an image of a larva. Larvae take 3 to 5 years to develop, and during this time they live on the riverbed, hunting other invertebrates. As you can see in the image, they have a nice streamlined, flattened body shape, and they're also covered in hairs, which helps catch sediment in order to camouflage them. So the aim of the Clubtail Count project was to increase our understanding of the species distribution. As a near-threatened species, it is a priority for us, and we want to be able to keep a track of how the species populations are doing, whether they're increasing or declining, and also by locating the populations and knowing where they're breeding, we can also put safeguards in place to help protect them. Now, our historic records for the species were pretty sparse, and this really reflects their elusive nature, with the adults spending most of their time hidden away in woodland, which makes them quite difficult to record. So the easiest way to identify the location of breeding common clubtail populations is actually to look for their exuvia. So when a dragonfly larva is ready to transform into an adult, the larva will crawl up out of the water onto the riverbank or nearby vegetation and shed its larval skin, transforming into an adult and leaving behind the larval skin, which is called the exuvia. And as you can see, it's got quite a distinct shape with almost a heart-shaped face, and there's not really any other species in the UK that looks like this, so they're quite easy to identify. Here we have a video of one in the process of emerging. This meant the survey was really easy to carry out and members of the public could easily get involved. You didn't need any experience in dragonfly recording or identification. And we had people from all walks of life getting involved, including a canoeing club from Oxford. And we advertised on social media through BDS magazines, also advertised with the help of other wildlife organisations such as the North Wales Wildlife Trust. And by the end of the 3 years, over 180 volunteers had gotten involved across England and Wales. So the Clubtail count focused on rivers with historic records. In Wales, this included the Vyrnwy, Tyfi, Tawy, Dee and Wye, as you can see on the map. Volunteers adopted 1 kilometre OS grid squares, also known as monads, and they were asked to search them 3 times during the Clubtail's main emergence period, which runs from late April through to the end of July. So I'm going to quickly go through the result maps for Wales. Black dots indicate the location of historic records from before the Clubtail count, so pre-2017. Green squares indicate monads which had no previous Clubtail sightings but had Clubtail sightings during the Clubtail count, so they were site gains. Red, orange, yellow sites are sites which had historic sightings, historic records, but where no Clubtail were found during surveys during the Clubtail count. And the bluey, pinky squares indicate sites or monads which had historic records and also had records produced during the Clubtail count project. Oh, and the grey squares indicate monads which have no historic Clubtail records and where no Clubtail were recorded during the Clubtail count surveys. So as you can see, the results for the Tyfi and the Tawy weren't fantastic. Of the 7 monads that were searched on the Tyfi, none produced Clubtail sightings during the project. If the population has become extinct on this river, which it looked like it has, then it'll be hard for it to become repopulated due to the kind of isolated nature of the river in relation to other Clubtail populations. On the Tawy, only 4 monads were searched. We would like to search more in the future, but of these 4, only 1 produced Clubtail sightings. 2 exuvia were found in the pink square in 2018. So while the population appears to still be there, it might be quite restricted. More survey work is needed. Over on the Wye, results were a lot more promising. As you can see, the Clubtail appears to be doing very well down near Monmouth. On the Wye, all monads that were searched down there produced results. Further up, 3 monads were searched on the Lug. There are historic records for the Lug just upstream for where it joins the Wye, but unfortunately none were found during the Clubtail count. And Clubtail were found as far north on the Wye as Hay-on-Wye, just on the English-Welsh border. Here we have the Vyrnwy and the Severn, and all monads that were searched on the Vyrnwy produced Clubtail count sightings. The Vyrnwy is connected to the Severn, which produced very strong Clubtail count results. Clubtail were found all the way from the Vyrnwy down south past Shrewsbury, all the way down to Tewkesbury. So the species is very well distributed on the Severn. Unfortunately, there wasn't a survey done on the Welsh border, across the Welsh border for the Severn, so that's an area we need to target in the future. Results were pretty good on the River Dee too, with Clubtail found all the way from Bangor-on-Dee, north past Wrexham, right up to Chester. And there was also sightings to the north of Wrexham on the Allen, so this is a river that would be good to investigate further in the future. So if you're wanting to spend a little bit more time looking at the results for England as well as Wales, please go and download the report. You can also download all the records from the survey on a Excel spreadsheet, and that's all up there as well on the BDS website. Here's the website address, and you need to go to Clubtail Count, which is in Other Projects in the recording menu. All the records for Clubtail are also on NBN Atlas as well, so you can download them there. So the report has now been circulated with wildlife and environmental charities and organisations, as well as local authorities, with the hope that it will be taken into consideration with regards to riparian and river management activities, as a number of insensitive management practices, as well as environmental issues, can have detrimental effects to common clubtail populations. And I'm going to go through some of those now. Water pollution is, of course, a big one, as their larvae are living on riverbeds, so they are sensitive to chemical and pesticide pollution, so runoff from farmland use of pesticides, eutrophication from nutrients runoff from farmland, as well as if there is a high sediment load that can effectively smother the larvae. So water quality is very important. Habitat destruction, so the destruction of riparian deciduous woodlands. So clubtail aren't terrible at dispersal, they're quite good flyers. So the woodland doesn't need to be directly next to the river, but it does need to be easily accessible. It needs to be close by. And obviously there needs to be areas of woodlands near to suitable breeding rivers for the adults to forage in. River reprofiling can have drastic impacts on clubtail populations. So clubtail larvae like slow-flowing water and rivers with nice, gradually declining gentle banks. So reprofiling and canalisation to create steep sides and increasing water flow can have really negative impacts on the habitat suitability of a river. And of course they can directly impact the larvae as well, so kind of wide-scale dredging or kind of bank management activities with machinery can directly kill off large numbers of larvae and really have an impact on the amount of dragonflies that emerge. Disturbance is a major threat, particularly to emerging clubtail larvae. Clubtail larvae don't actually climb that far out of the water before they start undergoing emergence. Usually they're less than 30 centimetres above the water surface. This means that they can be knocked off or damaged by waves caused by passing boat traffic. If you have high levels of boat traffic during the emergence period, it can have a highly detrimental impact on the number of common clubtail that successfully emerge. The emergence of common clubtails is synchronous. This means a population will all emerge during a relatively short period of time. If you have invasive bank management activities, such as mowing, this can have a highly detrimental impact and destroy large numbers of emerging adults. Gomphus vulgatismus is a unique and charismatic insect and worthy of safeguarding. In addition, conservation action that is beneficial for common clubtail can also benefit a variety of other wildlife. For example, the North Wales Wildlife Trust have been planting areas of black poplar, an endangered species of tree, near the River Dee. Not only will this be beneficial for common clubtail by providing adults with foraging habitat, it will also benefit a variety of other insects, such as moths, as black poplar is the food plant for many species of caterpillar. So, if this presentation has got you enthusiastic for dragon hunting, please visit the BDS website and visit the records section for more information on our upcoming dragonfly recording activities. If you live in the east of Wales, please keep an eye out for our white-legged damselfly investigation project. We'll be asking you to keep an eye out for this elusive species which again, like the Common Clubtail, is under-recorded. If you have any questions about Clubtail or questions about dragonflies in general, feel free to get in touch at conservation@british-dragonflies.org.uk. Thanks for listening.
Thank you, Eleanor. Really fascinating presentation there. We will take some questions after the next presentation, so if you